In this session, we will try to understand development of female gametophyte, which is commonly known as your embryosac, and the total developmental process is otherwise known as your megagametogenesis. So before going to megagametogenesis, we have to focus about the previous classes, some of the topics. That means in our previous classes we already studied about the structure of ovules and some of the basic features of the ovules. Today we are going to study about the megagametogenesis. So in this part we just study the middle part, that means the nucellus part of the ovule. We already studied the basic things, so we have not panic about the total structure. Just focus about the middle part, that means your nucellus part. So your nucellus part is consist of a number of diploid mass of cells, and the upper part of your ovule is otherwise known as your chalazal end, and the lower part is otherwise known as your micropylar end. So a single cell of the nucellus, that means the diploid mass of cells, compact mass of cells are going to divide, and a single cell is going to form your MMC, that means your megaspore mother cell. So your megaspore mother cell is again divide to form your next structures. So before going to the total mechanism, I want to clear a points to you that whenever you try to draw these types of diagrams of your megagametogenesis, try to mention the two parts, that means your micropylar end and chalazal end, whenever you draw the diagrams, because these parts are quite important. That means the micropylar end and chalazal end is going to differentiate the total structure. That means the micropylar and chalazal end going to differentiate the place of that part. So whenever you draw, remember that you have to mention the points, that means the micropylar end and chalazal end. So the megaspore mother cell is formed. Next to the megaspore mother cell, it is diploid in nature, that means 2n in nature. Nextly, the meiotic division takes place, that means haploid cells are going to form. So a megaspore tetrad is formed, that means these are n in number. 4 number of cells are going to form, which are n in number, and this is your micropylar end, this is your chalazal end. The cells are arranged in linear nature, that means linearly arranged cells are formed. Nextly, the linearly arranged cells, the cells which are present towards your chalazal end, that means the upper threes get degenerated, that means these three are going to degenerated. Upper three are going to degenerate, that means which are present to your chalazal end, and a single one which is present to your micropylar end is surviving, that means this is your surviving megaspore. Nextly, the surviving megaspore is going to form your embryosac, that means this is your basic points. From this, the formation of your embryosac is going to take place. So here a single surviving megaspore is developed. From this, the single surviving megaspore is going to differentiate, that means it is going to differentiate and a mitotic division takes place. Previously your meiotic division takes place, nextly your mitotic division takes place, and in this mitotic division, three number of mitotic division takes place in every single point. That means in every mitotic division the karyokinesis takes place. Before going to the mitotic division, I want to clear about your karyokinesis. Karyokinesis means the process in which the nucleus is going to divide. That means in every stages mitotic division takes place, but the karyokinesis takes place. That means the the nucleus is going to divide. So a single nucleus is there. First mitotic division takes place, and as I tell you that the karyokinesis takes place in first stage, so the nucleus is going to divide, so it is divided into two types, that means two nucleus are going to divide. Nextly, mitotic division, second stage of mitotic division takes place, 2 to 4 number of nucleus are going to divide. Nextly, third mitotic division, 8 nucleated cells are going to divide, that means 8 nucleus are going to divide, that means karyokinesis stopped here. Nextly, cytokinesis takes place. First is your karyokinesis, nextly cytokinesis. So before going to cytokinesis, the 3 cells, that means 8 number of cells are going to divide, 3 and 2, 8 number of cells. Nextly, the upper 3 are
going towards, these 3 are going towards your chalaza end, and the lower 3 are going towards your micropylar end, and 2 are present in your middle stage. That means I change the color here. These 2 are present in your middle part, 3 are going towards your chalaza end, 3 are going towards your micropylar end. Nextly, these are the freely nucleus, and around this nucleus the cytokinesis takes place. That means the nutrient medium, the cells are going to develop. So here over here the 3 number of cells are going to develop. That means cytokinesis takes place in the lower stage also. That means in your micropylar end also the cytokinesis is going to develop. But the middle part, that means the middle cell of your micropylar end going to a little bit bigger. That means it is your egg cell, so it is going to little bit bigger. That means the cytokinesis is takes place over here. So in your chalaza end and micropylar end the cytokinesis takes place. Next to the cytokinesis, the total developmental structure, that means the total structure is fully developed. That means 3 are present toward your chalaza end, which is otherwise known as your antipodals. 3 are present to your micropylar end, which is otherwise known as your 2 are your synogytes, and middle one is your egg, and the total structure which is composed of your egg and synogytes is otherwise known as your egg apparatus. So the 3 parts, among the 3 parts, the middle one is a little bit bigger, which is otherwise known as your egg. The middle part is otherwise known as your polar nuclei. There is no cytokinesis takes place in your nucleus part, that means in nuclei. So this is all about your, that means your embryo sac. That means the formation of your embryo sac is already takes place. I again repeat the total structure. Here the microspore mother cell is formed. Next the meiotic division takes place. Next to the meiotic division, the 2 number of cells are going to divide into n number of cells. That means haploid divisions takes place, and these are present in your linear motion. That means linearly the cells are arranged. Next to the linearly arranged cells, the degenerating takes place. That means the cells which are present towards your chalaza lines, the 3 number of cells are going to degenerate, and a single cell is survived. That means it is a surviving megaspore. The surviving megaspore is again differentiate, and nextly 3 stages. In 3 stages mitotic divisions takes place, and every mitotic division there is a single karyokinesis process takes place. That means one mitotic division, that means your one karyokinesis. Second mitotic division, your second karyokinesis. Third mitotic division, your third karyokinesis. That means in every stages, in 3 number of stages, three times the nucleus is going to divide. So a single nucleus is there. Second stage, your 2 nucleus, that means a single is splitting into 2. Nextly 2 splitting into 4, and 4 splitting into your 8. That means finally your 8 nucleus are already formed. Next to the nucleus are going to arrange. So they are arranged in your chalaza end and your micropylar end. That means 3 are arranged to your chalaza end and 3 are going to your micropylar end. This is all about your nucleus division. That means your karyokinesis process. Next to the karyokinesis, the nucleus is going to divide. Nextly the cytokinesis takes place. That means the nutrient mediums are going around the nucleus. That means in your upper nucleus they are arranged, 3 are arranged, which is otherwise known as your antipodal cells, and your micropylar, 2 are, the sides 2 are otherwise known as your synogytes, and middle, the bigger one is otherwise known as your egg, and 2 remaining are otherwise known as your polar nuclei. This is the total structure of your embryo sac. Nextly 3 are antipodals and 3 are, that means 2 synogytes and egg consist of your egg apparatus and your polar nuclei. This is your complete diagram of your embryo sac. Next to the embryo sac, I also told you that it is otherwise known as your 8 nucleated 7 cell structure. Let's count how it is known, how it is the formation. That means how the name is formed, your 8 nucleated 7 cells. How it is known as your 8 nucleated. That means let's count the nucleus. Actually the nucleus is 1 2 3 4 5 6 7 and 8. This is your 8 nucleus. So it is otherwise known as your 8 nucleated. It is clear. That means 8 nucleated is already formed. Nextly 7 cells. Let's count the cells. After cytokinesis, this is your 1 cell. Change the color. This is your one cell, this is your second cell, this is your third cell, this is your fourth, this is your fifth, this is your sixth, and the outer is your seventh. That means ultimately 8 nucleus are formed and 7 cells are formed. This is your 7 cells, that means 3 to your antipodal, 3 towards your that means 2 your synergids and 1 egg, and the bigger one, that means the egg apparatus. Total apparatus is otherwise known as your cell, that means 7 cells. So it is ultimately concluded that it is your 8 nucleated 7 cell structure. So this is all about your development of female gametophyte. Hope you all are understood.